And today's class is for 11th. It is topic is Golgi bodies. What are Golgi bodies? And Golgi bodies is the next cellular organ for which today we are going to discuss something very much important regarding it. So this is for class 11th zoology subject Golgi bodies. What are Golgi bodies actually? So actually, this was a scientist whose name was Camillo Golgi in 1898. So, a scientist, namely Camillo Golgi, in 1898, he first of all he observed some densely stranded reticuloid structures with nucleus. He found some bodies which are densely stranded. When he stranded them, they were found very much densely stranded inside the cytoplasm of the cell, and those bodies were having nucleus in them. And later on, these densely stranded reticulate structures with nucleus, which were first of all observed by Camillo Golgi in 1898, they were later on named as Golgi bodies by the later researchers. And further, they are formed of many flat disc-shaped sacs or cisternae of 0.5 micrometers to 1.0 micrometers in diameter. Means these Golgi bodies are formed of flat, flat disc-like structures, flat disc-like sacs or cisternae. These disc-like sacs are also known as cisternae and whose size range from 0.5 micrometers to 1.0 micrometers. Mostly these Golgi bodies are possessing these disc shaped sacs or cisternae of this size, that is 0.5 micrometers to 1.0 micrometers. And students, they are piled parallel to each other. Varied number of cisternae are present in a Golgi body. In a Golgi body, the exact number of cisternae are not found. One Golgi body may possess a one number of cisternae and other Golgi body may possess other number of cisternae. But you have to remember here they are piled together, they are situated in them parallel to each other. In a parallel arrangement, these cisternae are lying with each other in a Golgi body. And the most important thing here you have to remember is that these disc shaped sacs or these cisternae or Golgi cisternae, they lie near the nucleus with two faces. These Golgi bodies cisternae, they always lie near the nucleus and have two faces. One face is known as cis face or forming face, and another face is known as trans face or maturing face. And I will show you after some time the diagram, and where you will get a clear image of cis phase and trans phase of Golgi cisternae which are lying near the nucleus. This is an important aspect regarding this Golgi cisternae. And you have to remember that they are entirely different from each other but are totally interconnected. These two phases, they are totally different from each other. They don't resemble with each other, but they are very much interconnected with each other. They are not separated, but they are interconnected because these are the two faces. I am trying here to show you that suppose this is the nucleus and these are the Golgi bodies and these are Golgi cisternae or sac like disc shaped vesicles are cisternae, they lie in a parallel arrangement with each other. This one, this one, this one, this one. And these Golgi cisternae, they have two faces, which I told you. This one is cis phase, or it is also known as forming phase. Forming phase means here something gets formed, and when it enters into this cis phase, it is then transmitted through these Golgi cisternae towards this trans phase, and this trans phase always faces towards cytoplasm, and it is also known as maturing phase. Maturing phase means whenever a thing starts to form, it enters here in cis phase of Golgi bodies and move towards the trans phase. And on reaching to the trans phase or maturing phase, these things they got released into cytoplasm. Hence, this end or this phase is also known as maturing phase. And this is our nucleus. And endoplasmic reticulum always lie near the nucleus. And here the size of these Golgi cisternae, it ranges from 0.5 to 1.0 micrometers, and these are the Golgi cisternae. This is nucleus, and all you have to keep in your mind regarding the structure. And this is cis phase and this is trans phase, and these two phases are very much interconnected to each other, but they are very much different from each other. So let's move ahead. Golgi bodies help in packaging the material to be delivered either to intracellular sites or outside the cell. Whenever anything is needed to be taken, to be transported, to be delivered inside the cell or outside the cell, it is these Golgi bodies which help in packaging of that very material. So this is the foremost and important function of Golgi bodies in a cell, that it help in the packaging of material which is to be delivered either inside the cell or to the outside environment of the cell. Further, any material in a vesicle at the endoplasmic reticulum gets packaged with the cis phase of the Golgi body, and it then moves to the trans phase. That is why they lie in close association with each other. This means that if these are the Golgi sternae and here lies the endoplasmic reticulum, and as you have in previous topic you have read that endoplasmic reticulum is also made of tubules, cisternae and vesicles, spherical or rounded vesicles, and whenever anything is needed to be packaged from these endoplasmic reticulum, these vesicles, they are packaged into it, and these vesicles enter into these Golgi bodies or Golgi cisternae through these cis phases or forming phase, and then those vesicles are being transported towards the trans phase or maturing phase and then released in the cytoplasm. So there lie always in a close association of endoplasmic reticulum and this Golgi bodies, because both of these cellular organelles they help in the transportation of the things, in the packaging of things. So it is the vesicles of this endoplasmic reticulum which package any material inside them, and those vesicles then enter into these Golgi bodies at these cis phases or forming phases and then get transported towards these trans phases or forming phases, and then they are released into the cytoplasm. In this way, materials are being transported inside the endoplasmic vesicles from the cis phase towards the trans phase of the Golgi bodies. Hence, always these Golgi bodies lie in close association with endoplasmic reticulum inside a cell near the nucleus. So this is the close association of Golgi body and endoplasmic reticulum and the function of cis phase and trans phase. At any time you will be asked what are the functions of cis phase or and trans phase of the Golgi bodies. or why Golgi bodies always lie in close association with endoplasmic reticulum inside a cell. Let's move ahead. Golgi bodies are the important sites for the formation of glycoproteins and glycolipids. You must remember that whenever glycoproteins are formed, whenever glycolipids are formed, it is these Golgi bodies which are very much important sites for the formation of both these things. So, this is the next important function of Golgi bodies. Many proteins formed by the ribosomes in endoplasmic reticulum get modified first in Golgi bodies and then are released from its trans or maturing phase in cytoplasm. So, as you know, ribosomes play an important role in the formation of proteins, and when proteins are being formed from ribosomes in the rough endoplasmic reticulum, as I have already told you yesterday, what are rough endoplasmic reticulum and what is smooth endoplasmic reticulum. So, the endoplasmic reticulum, which possess ribosomes, which are granular, which possess granules of ribosomes, they help with the formation of proteins, and first of all, these proteins are being modified in these Golgi bodies, and then by entering into the cis phase, and then finally they are released at the trans phase or maturing phase into the cytoplasm. So this is the next important function of Golgi bodies, and this is it is rough structure which I have showed you already, and these are its two phases, cis phase and trans phase, or forming phase and maturing phase. Hope you will have understood this topic very much uh, deeply, and this is an important question which is mostly asked in the annual exams, Golgi bodies for class 11th, and this is your zoology sir. For any confusion, please feel free to contact me. Thank you. Have a good day.